So thanks to the organizers for having me at this workshop. I'm really excited to be here. So I'm going to tell you about interaction-based methods for uncovering genes that are functionally important in cancers. And this is joint work with Dario Gersi and Borislav Hristov. So those of you who know me know that we've been thinking about protein networks for a decade or more. So our work is driven by the observation that cells function on the basis of various types of molecular and macromolecular interactions. These can be interactions amongst proteins or proteins and small molecules, proteins and DNA, proteins and RNA, and other types of interactions as well. So if we want to understand how any cell works, whether it is a normal healthy cell within our body or a disease cell, we've got to have some sense about the underlying networks. And of course, this is only amplified when you think about things at the organism level, because these interactions within cells, you have cells that interact with each other to form tissues, organs, and organisms. So as a result of this, my group has been developing methods over the years for kind of two complementary problems. On the one hand, we develop computational methods for predicting various types of interactions, specifically various types of protein interactions, whether it's protein-DNA interactions, protein-protein interactions, or protein-small molecule interactions. And on the other side, we've been developing a variety of methods for trying to analyze the large-scale interaction networks that have emerged from high-throughput studies. When we think about trying to develop methods for predicting interactions, we often take a structural point of view. So can we leverage information about protein structure to make predictions about interactions or interaction specificity? And on the other hand, when we develop methods for analyzing protein interaction networks, we often take graph theoretic views. And so this sort of dichotomy on the one hand between structure and interactions has driven my work, and it also kind of framed the talk I'm giving you today. So often in the world of network biology, when you think about a network, you think about a single network for an organism. And this has sort of been traditional because of the type of data that's there. But of course, networks are dynamic entities. Even across different cell types within the body, you have different underlying networks. But this is especially true when you look across individuals. And so we have become interested in the last few years in trying to develop methods for uncovering how various types of networks vary, and also for analyzing this variation across networks. So if you look at humans, we of course are vastly different, whether on this slide or in this room, and it's really the networks that underlie us, in particular regulatory networks in this case, that drive the phenotypic change. And so this is how I got interested in thinking about cancer genomics. So as we've already heard about this morning, there have been large-scale efforts to sequence the genomes of individuals' cancers. And through the types of algorithms we heard about in the first talk, we've uncovered the alterations that are found in the tumors versus the genomes that are inherited. And this has been done across large numbers of individuals, and the interpretation can sometimes be done at then the population level. So there are many interesting questions. I know we'll hear a lot about them this week that you can try to answer with this type of data. But I was just going to focus on one type of goal, which is which of the numerous mutated genes, so looking just at mutations, single nucleotide variations within genes that encode proteins, play a role in either cancer initiation or progression. So trying to develop methods that take protein networks and protein interactions into mind to try to uncover which of the many altered genes are the ones that are really important for cancer. So I'm going to tell you about two projects. And the first one is joint work with Dario Gersi, who's now on the faculty at Nebraska. And here we're going to take a structural perspective. And we're going to focus on mutations that affect protein interactions in cancers. So I think it goes without saying, but I'll say it anyway, that mutations that affect protein interactions are critical. So if we look at a gene that encodes a protein, not all mutations are created equal. Those that interact with the functionally relevant molecule of the protein are going to be more important. So this could be the residues that interact with small molecules. This could be residues that are in a protein-protein interface, or this could be residues that are in contact with DNA, amongst others. And so our work begins with the idea that disrupted protein interactions can play a role in acquired disorders such as cancers, and of course also in inherited disorders. So with this in mind, we wanted to try to develop a pipeline that would identify residues that take part in interactions. And so what we did was we looked at variants with respect to some reference sequence. We mapped these variants onto the reference sequences. And then for each reference sequence, we compared it to other sequences for which structures had already been determined. If two sequences are similar, they tend to have the same structure. So if we found another sequence with which it had sequence identity greater than 60%, then we used its structure as a template for this sequence. So we required a sequence similarity of 60%. We used this great resource that's called BioLip, which has about 60,000 structures in it. And this is a partly curated database where binding residues have been identified. So within a protein structure, residues that interact with either nucleic acids, small molecules, ions, or peptides have been identified. So once we found a sequence that's similar to some structure in this database, we can transfer information about which residues are the ones that are taking part in binding interfaces. So this type of pipeline is automated, and you can download it at canbind.princeton.edu. And I wanted to say that with this structural approach, which is actually fairly conservative with respect to how much sequence identity is required, we can at current model about 22% of human protein. So it's not modeling all the human protein, but aspects of it. So now we have this pipeline, we can start asking some interesting questions. So for example, we can just look across healthy populations and look at the SNPs that are found across those populations. And so we can look at this in aggregate, and that's what we'll do to start with. So we can ask how many sites have structural information. So I said 20% of the proteins, and then a subset of the residues within these proteins. How many of these sites are polymorphic? How many of these sites are involved in binding using the pipeline that I just described to you? And then how many of these binding residues are polymorphic? And so here we're just going to consider missense mutations because we're taking this in mind with respect to protein structures. So what we found is if you look at the total number of sites that have SNPs, you get some number. And you can make a theoretical distribution that comes from this data, and that's just a hypergeometric distribution. And what you observe clearly is, in aggregate, polymorphic sites across humans tend to avoid binding residues. So binding residues tend not to vary across individuals. Now, that's not the case for each individual type of protein. So, for example, some proteins, their functions depend on their variations. So immunological proteins, for example, you would see a lot of variation, but just in aggregate, this is true. Okay, so then we can say, now what happens when you look across disease populations? And so here we looked at cancer missense mutations here across 8 TCGA cancer types. And again, we asked the same question. If we look at binding sites, how many of the residues have some mutation found within them across the sample of patients? And in this case, we actually see the opposite trend. so that we see that cancer missense mutations are actually enriched in binding sites as compared to what you would expect by chance alone. You can repeat this exercise instead of looking at missense mutations by looking at synonymous mutations instead. Now, if you have a synonymous mutation, you're not changing the amino acid, and so you're not going to have effect, at least on the structure. And in this case, across this same set of individuals, you see that there's no enrichment of mutations in the binding sites of these proteins. So the data I just told you about is an aggregate over 3 different types of binding sites, and that included nucleic acids, small molecules, and peptides. But if you separated them individually, you would see the same trends. So cancer missense mutations seem to be found preferentially within binding sites that are involved in nucleic acid, small molecule, or peptide interactions. Okay, so this was an aggregate analysis, and we wondered whether we could turn this around and make a per gene or per protein method to try to identify genes that are enriched in mutations in their binding sites. And I think this is an interesting way to go because kind of the one observation is that frequently mutated genes that are known to be playing a driving role in cancers seem to, by and large, have been identified. And so here's a plot looking at 22 different cancers. And it has 5 well-known cancer players along with 2 controls, and you can see large numbers of mutations across them. So I think the real challenge in this game then is in identifying less frequently mutated genes that play a functional role in cancers. And so we're hoping that this structural method would be one way of accomplishing this. Okay, so what we did next was look at things at a per gene or per protein level. And very simply, it works like this. So you take in a gene, you identify the binding residues, so shown in black here. You tally the mutations in the gene across all your samples, and you compute some kind of mutation binding score. In this very simple example, we're just counting up the number of times it hits a binding residue. So it's 1, 2, 3, 4, 5, 6, 7. So that's just some score. You want to know if this score is more than you expect by chance. So you can permute all the mutations across the protein sequence. On this randomized data, you can compute a mutation binding score. You can repeat this numerous times to build a distribution. And then you can get a p-value for this binding score. Now, this is the outline of what we do. I just wanted to say that, in fact, we take structure more into account than this. And so when considering binding residues, we actually consider the fraction of heavy atoms that are in contact with the ligand. But the overall outline is as follows. So now we have a method for looking at things at a per gene way that tries to figure out whether binding sites are enriched in mutations. So next, we can order our genes based on how significant their p-values are, and we can try to do some assessment. So if we did that, where we ordered the genes based on p-value, we can ask whether known cancer genes are enriched in the set with the most significant p-values or not. And in fact, that's what we observe. So if we use the method I just outlined with missense mutations, as we look at smaller and smaller p-values, we see a larger enrichment in these known cancer genes. We can repeat that process just focusing on synonymous mutations. Again, these don't affect protein structure. And so you here see this flatter distribution, so you don't see the enrichment. So as an example, this is the type of thing you might see with our pipeline. So here's the protein dicer, actually a domain of it. This protein, as most of you know, is involved in microRNA processing. When you look at endometrial cancer and glioblastoma, what you observe is that there are a number of mutations that you see that are involved in binding to a magnesium, that are involved in binding magnesium. And these mutations take this negatively charged amino acids, glutamic glutamic acid and aspartic acid and change them to amino acids that aren't negatively charged. And this negatively charged valley is required for catalytic activity. So it gives you perhaps a functional way in which these mutations are affecting the functioning of these proteins across these cancers. And so you can see there are other mutations as well. Those are shown in blue, and those are the ones that are not involved in binding, but these mutations are enriched. You can do this type of analysis across cancers individually, and what we've shown here is a heat plot where once you do an FDR correction, you identify a number of genes that are significant across these 8 different cancer types, and in red are genes, these are the genes, and in red are genes that are known cancer genes. And what you can see is many of these known cancer genes are actually enriched in mutations in their interaction interfaces. Many of these genes have these mutations across cancer types, but there are also other genes that have more cancer-specific patterns. Okay, so to kind of summarize this section of what I was doing, I wanted to say again that mutations within protein binding sites are under-enriched across healthy individuals, and they tend to be over-enriched in aggregate across cancers. And so this leads to a method that's based on the interaction interface for predicting genes that are functionally important in cancers, and we would hope perhaps in other diseases. We're doing a lot of ongoing work in this area, and in particular, we're trying to extend the applicability of this approach via predicted binding sites as well as protein domain information. So I think actually this is a very promising way to go. It turns out that 60% of human proteins contain at least one interaction domain. So we're going to have information about which of its residues interact with ligands. And I think we can push the applicability of our method by incorporating this other type of information. Okay, so for the second part of my talk, I wanted to, and this is joint work with Borislav Hristov with some input from my colleague Bernard Chazelle. So in this part of the talk, I wanted to talk about cancer mutations in the context of protein interaction networks. So very soon after the first large-scale protein interaction networks were determined, it became clear that these networks had a lot of structure within them. So these networks have this modular structure, and proteins that take part in the same complexes or biological processes or biological pathways tend to be proximal in networks. So, I think it's natural to think about interpreting the mutations that you see across cancers or across other disease types in the context of these networks. And so the goal with this type of work, putting together mutational information with network information, would be to identify groups of genes or proteins that together are mutated across individuals. Of course, this is something that's been proposed many years ago and in different contexts before cancer. So I think Roded worked on it with disease genes with Mendelian diseases. Ben has worked on similar things with respect to cancers. So this idea of interpreting mutations in the context of networks is clearly a natural one. And I think it's also clear why it would be especially useful in cancers because in cancer, so this is an example from the glioblastoma TCGA paper, they observed that there are mutated pathways, so 3 examples are given here. But when you look across the cohort of patients, you find that different mutations within these pathways are found in different patients. And so these occur in different patterns, as we heard about in the last talk. You can have rules, for example, or you can have trends of mutual exclusivity. So one key observation is that there are some genes that may be mutated in smaller fractions of the patients than others. Okay, so looking at mutations within the context of networks gives you a way to try to think about things at the level of pathways instead of individual genes, but may allow you to identify genes that play functionally important roles even though they're not perturbed across many cancer types. So the rough framework that we're thinking about is, in this toy example, is that you have a toy network, and you have a set of individuals, and you have a set of genes that are found to be mutated across these individuals. So what we do is we annotate each gene or protein with the individual where that gene is found to be mutated in. So for example, this gene is found to be mutated in patient 1, patient 2, and patient 3. And our simple goal would be to look for a small connected component that covers most of these patients, so that this small connected component shown in red, the patients would tend to have a mutation in one of these genes. And the idea is that these small connected components then correspond to these shared pathways that might be perturbed across patients. Now, I wanted to say this, of course, is related to previous work. I think the first that I know of is Ulitziet et al., so from Ron's group, but there's also been work from Teresa's group and Mehmet Koyuturk's group, amongst others. So our formulation is somewhat different, and so let me talk to you a little bit about the formulation. So in our case, we wanted to take a trade-off between the size of the subgraph that you're building and the overall number of patients that are covered. Right, so we don't ultimately require that all patients be covered. So if you choose all the proteins in your protein-protein interaction network, you're going to get high coverage of patients. You're going to cover all of them, or at least all of them that could be covered by the network. But you're not going to be uncovering anything causal, or that even less than causal, that plays a functional role in these cancers. On the other hand, maybe better, you can have fewer nodes in your graph. On the bad side, maybe you're going to explain fewer of your patient samples, but your connected component is more likely to consist of genes that are functionally related to each other. So the way we formulate this problem is to try to find a connected subgraph, G prime of G, that minimizes this objective function, which consists of two parts. One is a component that corresponds to the fraction of patients that are uncovered. That is, the graph doesn't contain a gene in which it has a mutation in. And the other is some measure of the size of the subgraph. And we don't know how important these two pieces are, and so we have this alpha parameter that kind of controls the trade-off. And though I won't talk about it today, I'll just say that you can solve this exactly with integer linear programming. The only trick you have to be careful of is that you need to make sure that your solution maintains a connected subgraph. But you can use different techniques to ensure that. In any case, you can solve this exactly, but it is time-consuming. So instead, we use a heuristic, a simple heuristic, which I'll tell you about next. But I wanted to talk a little bit about the size of the graph. So a very simple measure of the size of a graph would simply be the number of nodes, so the number of proteins that take part in your subgraph. This has the advantage of being simple and intuitive, but it has one major disadvantage, which is longer genes, on average, they're going to acquire more mutations. And so that's not really a good measure to use. And so we use a measure of size that's based on the weight, where this weight penalizes the gene for being longer and then rewards the gene for having more mutations. So we trade off here length and the number of mutations. So this is the inverse of the mutation rate. And then the size of the subgraph simply sums over these weights. So the heuristic that we use now to try to find this connected subgraph that minimizes this objective function where you're trying to cover patients and also keep your graph small is we start off by picking a node greedily, and we add it to the subgraph that we're building. And then we look over all the neighbors of the subgraph that we have, and we add to the subgraph the node that improves the objective function the most. So we actually allow hops of length 2, and that's why you may end up adding 2 nodes instead of 1. And then you just repeat this, and you stop once there are no more proteins that improve the objective. We've tested the exact optimization on small networks, and so it has similar performance to the exact optimization, but obviously it's much faster. So what I haven't said now is I have these two things I'm trying to trade off, and I use this alpha parameter to do the trade-off. How do I actually pick this alpha? So we could do it with some kind of magic, or we could try to take a data-driven approach to do this, and that's what we do here. So what we're going to do is we're going to vary alpha. We're going to withhold a fraction of the patients. On the set that's left, the training set, we're going to find G prime using the algorithm, the simple greedy algorithm that I just described. And then using that subgraph G prime, we're going to see how well we cover this withheld fraction, which is the validation set, with G prime. And then we just add one kind of layer outside of it, which is in the very beginning, just as a sort of a sanity check, we're going to withhold as a test set 20% of the patients. And then we're going to repeat this process I just described to you for choosing the best alpha on a training set and a validation set. And then ultimately, we want to make sure that your performance on the validation set is similar to that on your test set. Okay, and I won't actually show that data, but in fact it is. So this is a pretty robust procedure. So we ran this process on various TCGA data. I'm just going to show you today results on kidney cancer, where we looked at 416 patients. They had 22 mutations on average. We looked at various networks. The data I'm going to describe to you now is actually from a high throughput network that consists of around 8,000 proteins and 24,500 interactions. We've also looked at larger networks like BioGrid. Okay, so the first thing was picking an alpha. And so what we do here is we vary alpha in small increments, and we run our heuristic procedure, and we ask what fraction of the patients are covered. And we do this on the training set, and that's shown in blue. And that's what we're running our algorithm on. And then once we have something output, we're going to ask what fraction of patients are covered on this validation set, and that's what's shown in red. And so the idea is that while these two things are close together, we're having good generalization, and once these things get apart, then you're overfitting on your training set. And then we also track the number of genes in your subnetwork as you're running these different values of alpha. So in something like this, you can see that while we're at about alpha equals 0.5, we still do pretty well when using the training set, and then with respect to coverage on both the training set and the validation set. But when you have very large values of alpha, you get very large subnetworks, but you have a big difference between the validation set and the training set, and the coverage on the validation set doesn't improve. Okay, so this is how we choose alpha. So say we chose an alpha of 0.5. Now to actually use this network-based approach to try to predict the genes that we think are most likely to play some functional role in either cancer initiation or progression, we do the following. So we repeat many times. We randomly hold out a fraction of the patients. We run this heuristic procedure. So this procedure already has randomness built into it, so we're not going to get back one solution. So it doesn't make sense to just do this once. We're going to run this heuristic procedure, and then we're going to rank the genes by the number of times it's output across these multiple iterations. So that gives a per gene score based on the algorithm that we just described. So now we can ask how we do. We've done different validations, but here's just an example where we look as we consider more and more genes by their score. We ask what fraction of the genes are cancer census genes. And so this interaction-based method I just described is shown in blue. We can implement the same thing, but not use any network information. And we just are calling it the set cover approach here. And of course, that's also been used by itself to think about disease genes, and that's shown in red. And so you can see across the range of considering more and more genes, including network information helps. And then we can also consider other approaches. So here we've just put one of the frequency-based approaches. It's MuteSig, but the recent version of MuteSig. And you can see that it dominates it as well. So using this type of interaction information is helpful. You can do other types of downstream analysis. I won't go into that, but the list of genes that you would get, if you look at those that occur in at least half of the output subgraphs, they're enriched in known cancer pathways, they're enriched in relevant Go terms, and they contain recently and newly implicated cancer genes. And with respect to future work, I think here we've really just considered missense mutations in protein coding regions, and trying to extend this framework to integrate other type of perturbations is where we'd like to go. And to stop, I told you about two projects, a structure-based project related to interaction interfaces, and a network-based project relating to thinking about mutations in the context of networks. And I wanted to thank Dario and Borislav, my collaborators, with this.
Okay, we have time for a couple of questions. Yes, Teresa.
So I was wondering, so one of the motivations to look to use network approaches, as you mentioned, is to discover some of those drivers that might be less frequent. Did you look looking at the drivers that you discovered, are there any that would be in this category that are really infrequent?
That are known drivers that are infrequent, or that are infrequent? Yes. So we also, I didn't show that, but we also get genes, for example, that aren't that have very low mutation counts. Sometimes that also aren't mutated. So, and some that are known to be drivers in other cancers, but aren't mutated in the cancer that we're looking at.
Yeah. So first question, you're trying to cover all of the patients, right? Would it make sense to maybe relax that constraint, because that might be producing some overfitting for patients where you might not?
Well, that's what this alpha parameter is for. So we have this alpha parameter that says that lets you cover fewer patients, because as you're saying, you really might overfit. And so a key part of the procedure is actually picking the alpha so that you ensure that you're not overfitting and just covering patients, even though it might not be relevant.
And then are there some sort of information theoretic measurements on the network rather than just purely size?
I actually think that the size part of this framework could be improved. I think often, I think that's something that we could think about in the future as well. I think what we're doing now, which is considering mutation rate, is a reasonable way to proceed, but perhaps you want to think about not just the number of genes that are in your network, but also things like the density of the subgraph that's induced or things like that.
Okay, last question up here.
It's very related. So you can see the size of the graph as a regularization of the graph.
Mm-hmm.
So it's like trade-off between the variance of the solution. And so maybe one first question, how stable is the solution? Because you repeat it many times and you average.
Yeah.
So what is the variance?
Yeah. So I don't think we've measured that directly. I mean, certainly at the top of the list, there's not a lot of change in terms of in the end the genes that are output. What changes is later on, and that's why we run it multiple times, and we actually use that to score the genes.
Okay, let's thank Mona once again.